Welcome back friends. In this session we are going to discuss few questions in NEET point of view from the chapter Molecular Basis of Inheritance. Let's go. If you see the first question, thymine can also be designated as. So what do you mean with thymine usually? So thymine means, that is a nitrogen base which is present in DNA, but you cannot find this thymine in RNA. Instead of thymine, you will be finding what? You can find uracil in the place of thymine in case of RNA. Whereas in case of DNA only, you can find this thymine. So what is the relation between the thymine and uracil? If you see the options, the four options you can see here, in all the four options they have given methyl uracil, but the only change is the 3, 2, 4 and 5. So what is the 3, 2, 4, 5 means? That is a position of the methyl group in uracil. In the ring structure will be having carbon, so that denotes the position of the carbon. If you see the structure, you can clearly identify the answer. You can see the blue color, that is uracil structure. In uracil, the fifth carbon has H, that is denoted in red color, whereas in thymine you can see here the fifth position carbon, the H is replaced by CH3. So CH3 means what? Methyl group. So methyl group is replaced in the fifth position. So the only difference is methyl group is present in thymine, methyl group is absent in uracil. So the answer will be the last option. You can see here 5 methyl uracil. So this is the answer. Why means methyl group is present in the fifth position. So only it is called as 5 methyl uracil. So thymine can also be called as 5 methyl uracil. If you go to the next question, in RNA, every nucleotide residue has an additional dash group present at dash. So you can easily identify the answer. So what will be the answer? So what will be nucleotide first of all? So nucleotide means that will be having what? Nitrogen base will be there, as well as sugar, that is ribose sugar or deoxyribose sugar. In the case of RNA, it is ribose sugar. And also you can find phosphate. So all three together it is called as nucleotide. So in RNA you will be having some additional group. What is that additional group? You can see the options, whether it is phosphate group or it is hydroxyl group. In the third position, second position is be given. You can see the third, like C and D options, they have denoted the prime 2 prime, whereas here they didn't say about the prime, just they denoted 2. So we should not go for that. So always the positions will be denoted like 2 prime, 3 prime or 5 prime, like that only the positions will be denoted. So what will be the answer? You can see here the structure you can clearly identify. First, in the case of DNA, you can see the blue color circle denotes the deoxyribose sugar. In the second position have only hydrogen, whereas in case of RNA you can see in the second position of the ribose sugar it has what? OH group. So what is the additional group present here? OH group. Where it is present? In the second position. So the answer will be D. What is D? OH group will be present in the second location, that is called as 2 prime. So the answer will be what? OH and 2 prime. So that is the answer for this one. So that is the difference here. So if you go for the next question, you can see a nucleotide has the following components. As I already said, it has 3 components. We can see here the first and last option they have given hexose sugar. So in the case of DNA, RNA, we don't have any hexose sugar. We have only ribose and deoxyribose sugar. So in this case, they didn't give about deoxyribose. They have given only ribose sugar. So in B and C, you can find this ribose sugar. Whereas the only change you can see in the second option, they have mentioned a phosphorus base and nitrogenous group. Whereas we will be telling that nitrogen base and phosphate group. Phosphorus group is not there. Phosphorus base is not there. That is phosphate group. As well as nitrogen group is not there. That is nitrogenous base. So answer will be C. Nitrogenous base, phosphate group and ribose sugar. All three together is called as what? Nucleotide. So the next question, if you go, before that you can see the structure of the nucleotide here. You can see, yeah, you can see here the adenine, that is a nitrogen base, plus that pentose sugar, you can see that is called as nucleoside. Nitrogen base plus sugar is called as what? Nucleoside. Whereas you can see when the phosphate group is attached to the nucleoside, it is called as nucleotide. So only base plus sugar is called as nucleoside. When the phosphate is attached to that, it is called as nucleotide. How you can remember? So only sugar is there, it is called as nucleoside. Nucleoside has a S. So S and S, sugar is there. Simply you can remember like that. So the next question, how many types of nucleic acids are found in living organisms? So from bacteria to higher animals, we have two kinds of nucleic acids. In case of virus also we have what? Nucleic acids. So we have two. What are they? DNA and RNA. So the answer will be what? You can see the structures. The DNA and RNA are the two nucleic acids present. RNA means ribonucleic acid. DNA means what? Deoxyribonucleic acid. So these are the two nucleic acids present in living organisms. No other nucleic acids are present. Only DNA and RNA. So if you move to the next question, you can see which nucleic acid in some virus can also act as genetic material. Yeah, this also I have discussed in some session. So in case of HIV, so HIV means what? Human immunodeficiency virus. So in that virus, the genetic material is RNA. But in some viruses, DNA can also act as genetic material. In some viruses, RNA can also act as genetic material. So in this case, the answer is RNA. Because maximum animals will be having DNA, but rarely the viruses, some viruses will be having what? RNA as their genetic material. Here mRNA, tRNA will be not becoming. mRNA, what do you mean mRNA? mRNA means messenger RNA. It carries a message from DNA and then it converts into protein molecule, that is mRNA. tRNA helps in the translation process, that is the formation of protein synthesis. In protein synthesis, tRNA will be acting as adapter molecule, mRNA acts as messenger molecule. So these two will not be the answer. So answer will be B, that is RNA. So RNA will be the answer, ribonucleic acid. So the next question, DNA is the polymer of. So usually what you will write, suddenly you will be writing after seeing the first option, you will be writing as nucleotides. But you should not write like that, because we have two types of nucleic acids. One is DNA, another one is RNA. So in case of, generally you can tell it as, in case of in case of nucleic acids, which is the polymer, it is made up of what? If they ask like that, you can write nucleotide. Generally if they ask in nucleic acids, you can write as nucleotides. But if they ask specifically DNA or RNA, you have to write in different manner. Suppose here they ask DNA. DNA means it consists of deoxyribose sugar. So you have to mention as deoxyribonucleotides. Suppose if they ask RNA, you have to write ribonucleotides because they have ribose sugar. Generally if they ask nucleic acids, you can write nucleotides. Whereas here the answer will be the D, deoxyribonucleotides. So deoxyribonucleotides will be the answer. So the next question, the length of the DNA is defined in terms of. So this also I have discussed in my session. So in case of DNA, the length can be defined. How? You will be writing as base pairs, but here you can see the options. First one, number of nucleotides, base pairs, and then both A and B and mu m they have given. So the answer will be C, that is both A and B. Why? Means in maximum animals, from bacteria to higher organisms, the genetic material will be DNA, that is double stranded DNA. In case of double stranded DNA, you will be having what? Base pairs. A pairs with T, G pairs with C. So we will be having base pairs. So we can measure the length with the help of number of base pairs in case of double stranded DNA. In some virus, you will be having what? Single stranded DNA as a genetic material. In those viruses, we can measure the length with the help of only nucleotides because they don't have base pairs. In case of single stranded DNA, only we have what? Nucleotides like A, T, G, C, like you'll be having what? Single strand only will be there. So no base pairs will be there. So in that case, we will measuring use nucleotides. So the answer will be both A and B. In case of double, specifically if they ask double stranded DNA, how we can measure the length, means that is base pairs. Without specification, simply if they ask general DNA, means you have to write both nucleotides as well as base pairs. Suppose if they ask generally RNA, means so you have to write what? Single stranded RNA will be there. In that case, nucleotides. Suppose double stranded RNA means base pairs. So based on the question, you have to answer. So if you go to the next question, you can see here, bacteriophage 5 into 174 has how many that nucleotides or base pairs, the genetic content. So the answer will be what? This bacteriophage 5 into 174, the genetic material will be, as I said in the previous question, so genetic material of this virus is single stranded DNA. So in case of single stranded DNA, we will not be getting base pairs. So A and D will not be the right answer. So it may be either B or C. So in your textbook it has been given, it is a direct question actually. So 5386 nucleotides. Why means here base pairs will not come because this bacteriophage has only single stranded DNA as a genetic material. So in that case they will be having what? 5386 nucleotides. So this is the genetic content of the bacteriophage 5 into 174. 5386 nucleotides because they have single stranded DNA. So moving to the next question, a nitrogenous base is linked to pentose sugar through what? So these also you can easily guess because in my session I have told you what are the linkages present in the nucleotides. Yeah, you will be having what? Between the nitrogen base and sugar will be having N-glycosidic linkage. To confuse you, they have given two options: O-glycosidic linkage, N-glycosidic linkage they have been given, but the answer is N-glycosidic linkage. That is between nitrogen base and the sugar molecule. Between the sugar and phosphate will be what? Phosphoester bond. Then where will be the phosphodiester linkage will come? When the two nucleotides are joining together. In dinucleotide you can find this phosphodiester linkage between 3' of first sugar and the 5' of the second sugar. You can find this phosphodiester linkage. You can see the structure here, you can easily identify. See here, that is the glycosidic linkage is present between the nitrogen base and sugar, the first nucleosides you can see there, that is glycosidic linkage, that orange color dotted around, whereas bottom you can see here clearly between the sugar and phosphate you can find what? Phosphoester bond. So these are the main two bonds here. Between the nitrogen base you will be having what? Hydrogen bonds. So these are the three major bonds present in the DNA structure: hydrogen bonds, glycosidic linkage, phosphoester. In case of like between the nucleotides we are having phosphodiester bonds. So that is the answer. So the next question, uracil is associated with which sugar? You can easily guess the answer here. Deoxyribose, sometimes ribose, only ribose, hexose. Where you can find this uracil? Only in RNA you can find this uracil. So RNA consists of which sugar? It has deoxyribose or ribose. It has only ribose sugar. RNA has only ribose sugar. So only in ribose sugar you can find this attachment of uracil. You can see here the uracil ring has been attached. You can see the uracil ring has been attached to the ribose sugar. In the second position you can see OH group is there, that is ribose sugar, so that is linked with the uracil. So uracil will be associated always with the ribose sugar. The next question, the 5' end of the ribose sugar bears what? Ribose sugar. What means ribose sugar? That you can find in the RNA. This ribose sugar will be having or will be present in the RNA. So what will be present in the 5' end? You can see the structure, you can easily identify the answer. See the first position, the base will be attached. Either it may be adenine, guanine, cytosine or uracil, because it is RNA, so uracil will be there in the first position. Second position will be having OH, and third position the another nucleotide will be coming and joining, that is OH, and fourth position will be having carbon, and fifth position CH2 will be there, CH2. So there only the phosphate group will be attaching. So in the fifth position of the ribose sugar you can find phosphate group, that is clearly mentioned here. So the answer will be phosphate group C. The next question, 3' end of the polynucleotide chain bears. This is polynucleotide chain, you have to see. What is a polynucleotide chain? So it consists of numerous nucleotides. So options are given like phosphate group, hydroxyl group, methyl group, nitro group. So you can see here, this is the polynucleotide chain. You can see clearly. Yeah. In the 5' and the red color arrow, so that denotes the first nucleotide, where the 5' it has phosphate group. So phosphate will be attached in the 5' end. So there only the phosphate will be there. Whereas the bottom you can see green color arrow, that is a 3' end. 3' end, the ribose sugar, third position will be having what? OH group. So always in the polynucleotide chain, one side 5' end will be having phosphate group, 3' end will be having hydroxyl group. So the answer will be B, hydroxyl group. Suppose if the question is 5' end, the answer will be phosphate group. So the remaining questions I will be discussing in the next session. Thank you friends. Thank you for watching this video.